Hello, everyone. This is Anne, practitioner and genetic specialist here at Connor's Clinic. And in this video, we are going to talk about iron and the Fenton reaction and the genes in your genetic report that are related to this. So when we're talking about iron, we are talking about the mineral that is necessary for proper bodily function, important in many things that we need for a healthy body. So iron is involved in the production of heme, which then in turn is involved in producing hemoglobin. And hemoglobin is the molecule within the red blood cell that carries oxygen from the lungs and delivers it throughout the body. So we can see right here, it's talking about the iron, the health benefits of iron. We see the formation of red blood cells. So the red blood cells, again, they contain hemoglobin, and hemoglobin is what carries oxygen throughout the body. So we need, obviously, to have oxygen throughout the body. It is important for cognitive function, important for strong immunity. Iron is going to help prevent anemia. So anemia is low iron conditions where all of these things are not happening properly because we have low iron. It's involved in energy production and, really the ability to use our muscles. If we have anemia, we have low iron, we have, can have brain fog, we can have fatigue, we can have really sore muscles. It's just hard to move. And then iron is also involved in helping maintain a good mood. So iron is very important for health. And we have to get iron from dietary sources. And so here is an image that's just listing some of the dietary sources of iron. There's plant-based sources and then animal-based sources. And really there is a difference between animal and plant-based iron. So animal sources are going to contain that heme iron that is better absorbed and utilized than plant-based sources of iron. But it's abundant, in leafy greens, red meat, chicken has some, broccoli, spinach is high in iron, pumpkin seeds. So lots of different foods have a great source of iron. All right. And then we need to talk a little bit about kind of the different forms of iron. So there is bound iron and there is free iron. Bound iron is when the iron is bound to a molecule that is kind of keeping it from being in a chemical state where it is reactive. So an example of bound iron would be the hemoglobin. So iron is bound to that hemoglobin within the blood, so it's not chemically reactive. And then another example would be ferritin. So within the cells, ferritin is holding on to that iron. So when it's bound, it's being held until the body needs to utilize those sources. So that is the proper way we want to have iron within the body. It's bound to these protein molecules, and then when we need it, it is facilitated and then used appropriately. And if that is not happening, then we can have free iron. So free iron is unbound and that's where it can be reactive, chemically reactive, and it can be damaging to the body, especially if it is in excess. If we have excess amounts of this unbound iron, it can create damage. Short term, your body has checks and balances to keep that in check, keep it in place from the damage getting out of control. But long term, if we have an excess amount of this free iron, it really can lead to damage to the cells, damage to tissue, and damage to organs. And so we have up here on the screen kind of an example, a visual example of what happens if we have this free iron that's causing this stress or oxidative stress within the body. So, you slice an apple, and you've eaten some of the apple, and you walk away and you come back an hour later to finish up that apple. And what happens? Well, the apple has turned brown. That is oxidation, and that is essentially what happens if we have that free iron, unbound iron within the body. It creates that oxidative stress and really kind of damages things that it comes in contact with. So this is just a little visual example of oxidation. All right, and here are some examples of causes of excess iron. So, as we're talking about genes, we can have genetic predispositions, variants in our genes that, kind of give us a predisposition for excess unbound iron. So the damaging effects of this iron, and we'll talk about some of those. Alcohol consumption can increase iron absorption, excessive red meat consumption, cooking in cast iron skillets. So when we're, warming up our cast iron skillets and cooking food, some of the iron can leach into the food, especially acidic foods like tomatoes. So we need to be careful of that. If we have a predisposition for this. And then there's processed foods that are fortified with iron. And a little note here that says this form of iron is inorganic and can be much more dangerous than iron that is naturally occurring in food. So really, we don't want to be eating a lot of packaged foods, obviously, in the first place. And certainly we don't really want foods that are fortified with inorganic iron. Supplements obviously can contain iron, so it's good to know, good to look at these genes and see, okay, do I have a predisposition for improper facilitation of this iron and potential inflammation or damage from excess iron? So we wouldn't want to be supplementing with iron. And then it's also important to know what form of your iron supplement you are taking, because there's some good forms and bad forms that we want to watch out for. Iron synergists that can increase absorption. So an example of this would be vitamin C. So vitamin C increases the absorption of iron. So if you're eating red meat and you're drinking a big old glass of orange juice or something, that vitamin C from the orange juice is going to allow for excess absorption or more absorption of that iron from the red meat. So something to be aware of. And then hepatitis or abdominal injuries can, create some excess iron as well. All right, so when we're looking at genes involved in iron regulation within the body, these are some of the ones that we're looking at within your report. The HFE gene is really a gene that is involved with a diagnosis of hemochromatosis. So that is an iron overload condition, so that excess absorption of iron can create excess free iron. So we want to watch for that. And then these are genes that are involved in, so this would be the HFE gene would really be excess free iron within the blood. And then these genes here are kind of relating to ferritin. So variants here can increase absorption of iron into the cells. And we need to watch for that. This gene here is the only gene, the SLC40A1 is the only gene that exports iron from the cell. So we have a number of ways that iron can get into the cell, but we've only got one exporter out of the cell. And so if we have variants here, then it can lead to too much iron within the cell, which is obviously something that we need to be careful of. And then this last gene listed here, this CP gene, that is the gene that creates ceruloplasmin. This is necessary for healthy use of iron. And production of ceruloplasmin is dependent on vitamin A. And so when we know that, then we also need to look at, we have a gene that is involved in vitamin A, the conversion of beta carotene into the active form of vitamin A. That's the BCM01 gene. So if we have issues in the BCM01 gene, that can also affect this CP gene as far as ceruloplasmin and then its involvement with healthy use of iron as well. All right, and we have this nice graphic here from Nutrigenetic Research Institute. So this is where, the certification course that we went through, our practitioners here at Connors Clinic. So this is the training course that we did. And so this is just, one of the ways so that excess iron can, negatively be used to create some of these damaging things and inflammation. So this here represents the superoxide free radical, which is a free radical that is, it's the most abundant free radical within the body. It's produced by normal metabolic functioning of your cells. But we have other genes in place to neutralize this. So again, that checks and balances. So your body is going to create some superoxide, and then we have this SOD gene here. This gene is the superoxide dismutase gene. So it's going to take that superoxide and convert it into hydrogen peroxide. Then this hydrogen peroxide needs to go another step farther to be neutralized. We need the catalase gene. Some other genes are involved as well. Ultimately, we're at a neutral point with water and oxygen. So that is desired. When we have this excess free iron, this unbound iron, the hydrogen peroxide can bind with iron, and the two together can create these hydroxyl free radicals. And this is what is contributing to that oxidative stress that is damaging cells, tissues, and organs. And when we have production of this, if we have excess production, it then, triggers other genetic pathways to kind of try to balance that, but in the process can create some other damaging oxidative, things here. This peroxynitrite is oxidative as well, can contribute to cardiovascular issues, and lots of, kind of tissue damage, pain that might migrate from one place to the other. You can't quite pinpoint, can be due to excess peroxynitrite. So this hydroxyl free radical can stimulate your NOS gene, that's nitric oxide synthase, that produces nitric oxide. And excess nitric oxide is not good either. So if we have excess hydroxyl free radicals, we could be ending up creating excess nitric oxide, and then we can be creating peroxynitrite. So lots of negative things can happen when things are out of balance. So that is the beauty and the value of looking at these genetic pathways to see where we might have some of these weaknesses, so we can correct these things from a genetic, molecular level, so everything can work properly, and we're not going to run into conditions down the road. So it's good to look at these genetic pathways. So hopefully this helps you gain some understanding about iron, the benefits of iron, but then also the negatives if there's excess iron, and the inflammation that can develop if you have some excess iron. All right. Well, thank you for listening.